Hi, my name is Steph Varghese, and I'm an undergraduate researcher of plant ecology in the Wright Lab at Cal State LA. I am honored to be sharing with you today some insights into how the interactive effects of climate change might impact plant community composition in the near future. The functioning of ecosystems, as well as their capacity to supply vital services, is contingent upon primary producers' abilities to withstand the direct and interactive effects of climate change, such as warming and drought. Previous work has shown that plants can respond differently to warming and drought depending on their functional traits and photosynthetic strategies. For example, as demonstrated by Cowles et al. 2016, cool season C3 grasses, which are generally less drought tolerant than warm season C4 grasses, can experience disturbance from the drying effects of warming, while warm season C4 grasses, which are generally better adapted to drought, can actually experience positive effects from warming. However, what we are still lacking is a more detailed, longer-term and larger scale understanding of which plant functional groups will be favored under warmer and drier climates. Indeed, global temperatures have been increasing for decades. According to the National Oceanic and Atmospheric Administration, the global surface temperature this past January 22 was 1.6 degrees Fahrenheit, or 0.89 degrees Celsius, above the 20th century average of 53.6 degrees Fahrenheit, or 12.0 degrees Celsius. But we aren't entirely sure what the interactive effects of warming combined with drought will mean for plant communities worldwide. One theory suggests that warming could amplify the negative effects of drought upon primary production. Another postulates that the two effects might instead cancel each other out. We funneled all that curiosity into one essential research question, and that was, how might elevated warming and drought alter the functional composition of plant communities in the near future? We hypothesized that grasses, particularly those that utilize the C4 photosynthetic pathway, would outcompete legumes and non-leguminous forbs in warming, drought, and warming combined with drought treatments. We predicted this to be the case because grasses are generally the most drought-tolerant functional group, with C4 grasses having the additional advantage of being heat tolerant. Our experiment took place at the Cedar Creek Ecosystem Science Reserve of the University of Minnesota. Cedar Creek is part of the Long-Term Ecological Research Network, which is a group of research sites funded by the National Science Foundation, tasked with addressing the challenge of studying ecological phenomena over long periods of time. Cedar Creek's Big Biodiversity Experiment investigates the effects of plant biodiversity on ecosystem processes. Nestled within Big Bio is the Biodiversity and Climate Drought Experiment, otherwise known as BAKED. This is the sub-experiment that the present study focuses on. In BAKED, warming and drought treatments are imposed on 1, 4, and 16 species plots of randomized plant functional composition. Altogether, this comprises a full factorial diversity by warming by rainfall experiment. Plant species were randomly selected from a pool of these 19 perennial grassland species. Each baked plot features an unwarmed subplot, which serves as our control, an unwarmed subplot with rainfall removed, a heated subplot, and a heated subplot with rainfall removed. Soil drought is simulated through these rainout shelters, which exclude approximately 43% of precipitation, while air warming is implemented via these overhead heating lamps, which warm approximately +3 degrees Celsius. In early August of 2019, baked above ground biomass was harvested from 2.1 meter by 1 meter clip strips per plot. Next, the biomass was sorted by species, oven dried, and weighed. All data, including end of season plant functional composition, dry mass, and species abundance, were recorded in a spreadsheet format to be cleaned and thereafter analyzed in R. In this first figure, we have our drought and warming treatments on the x-axis and the log of grass forb proportional abundance as our response variable, which is a measure of whether above ground plant biomass was comprised mostly of grasses for y is greater than 1, or mostly of forbs for y is less than 1. Our 16 species plots under control, drought, warming, and drought plus warming treatments all contained a higher proportional abundance of forbs compared to grasses. In other words, all of these bars are below the y equals 1 mark. Importantly, although grasses never truly dominated over forbs, adding drought and heat stress significantly altered plant species composition in favor of grasses. This gave us a general idea of how the effects of climate change might alter plant functional composition. But then we wondered, might there be even finer scale effects at the individual species level? This is a question that we're still trying to answer. From this preliminary figure, we can tell that there is in fact substantial variation in the effects of warming and drought manipulations upon plant biomass at the finer scale species level. And while we are still trying to make sense of this novel finding, the general idea is that functional category alone is not always an infallible indicator of species success under warming and drought. Indeed, something really interesting is happening at the species level that is indicating that some species are not suited for a warming and drying climate and are going to be at risk in the near future. So, to summarize our findings, we first discovered that adding drought and heat stress can significantly alter plant species composition in ways that may effectively stabilize primary productivity in favor of the fittest individuals, which, in this case, turned out to be the grasses, which were better equipped than forbs to thrive under conditions of warming and drought. Then, when investigating the effects of climate change at the individual species level, we found that different species can have different responses to warming and drought based on factors other than their functional category alone. And this tells us that functional category is not always an infallible indicator of species success under warming and drought. Altogether, these results suggest that there remains a lot to be understood regarding how the cumulative effects of climate change will impact plant communities in the future. Future studies might want to explore how these climate-driven changes in plant community composition might affect interactions with pollinators. For example, how might treatment-induced changes in plant functional composition have cascading effects on insect pollinators via changes in forage? In addition, how might the diversity or abundance of insect pollinators in turn affect plant functional composition? Here are my references. And with that, I would like to thank my research advisors, Maggie Anderson and Dr. Amy Churchill, the Associate Director of Cedar Creek, Dr. Forrest Isbell, my supervisor, Sydney Hedberg, research coordinators Troy Mielke and Callie Worm, and all the dedicated and hardworking faculty, staff, administration, volunteers, and interns of the Cedar Creek Ecosystem Science Reserve.